Okay, so today we'll discuss about the microscopic structure of urinary system. Okay, mainly I will be focusing on the microscopic structure of kidney. So before I start, let me tell you one thing. The microscopic structure of kidney is a bit complicated. Whatever we have discussed in liver, okay, it is going to be more complicated than the liver. So I'll request everyone to listen carefully. Okay, and next week I will show you the slides of the kidney. Okay, before I start, I want to ask some questions. I hope you have seen the specimen of kidney, okay? Then you have studied the parts of the kidney, okay? How many surfaces, borders, poles, followed by the coverings of the kidney, okay? In the covering, I hope you have seen the renal capsule, okay? Next part, which you have not studied till now, that is the coronal section of the kidney. Okay, coronal section of the kidney, I don't think so you have studied. No. Okay, so don't worry, I will show you the coronal section of the kidney today. That is also important because this is the way how we can proceed to the microscopic structure. Unless and until you understand the coronal structure or coronal section of the kidney, okay, it will be a bit difficult for you to understand the microscopic structure. Okay, because this is the way to proceed for the microscopic structure. Okay, now everyone you know that in kidney nephrons are there, which are the functional unit of kidney. Okay, and you have studied also about the structure of nephron. Okay, so today we'll see that too, that also in detail, not in brief. Okay, now if you see the shape of the kidney, it is bean shaped. Okay, kidney is bean shaped, and where it is situated? It is situated retroperitoneally on the posterior abdominal wall. If you have seen the position of the kidney, okay, retroperitoneal, that also in the posterior abdominal wall. Next, the kidney which is present, it is covered by a connective tissue capsule that is called as renal capsule, and in that renal capsule, irregular collagen fibers are there, okay, and the capsule is mainly made up of two layers. So when you have studied the layers of the kidney, at that time you have noticed that multiple layers are there. For example, first is the renal capsule, then perinephric fat, then renal fascia, then paranephric fat. Okay, like these different layers are there which you have studied. Now I am asking you to concentrate on renal capsule, which is the innermost covering. Okay, now this particular renal capsule, okay, so the kidney is invested by a adhering capsule consisting of dense irregular collagen fiber. So if you see the renal capsule, it is adhered to the kidney. Okay, next, this particular capsule of the kidney is having two layers, outer layer made up of fibroblast and inner layer made up of myofibroblast. Okay, next, if you take the coronal section of the kidney, okay, just imagine the sections which you have studied earlier: coronal section, sagittal section, axial section. Okay, so what you have to do, you have to take the kidney and then you have to cut it. How you will cut? In coronal section. After that, you can open the kidney. Once the kidney is opened, what you can see two things. First is the reddish-brown cortex and inner pale medulla. If you remember the suprarenal gland, which we have discussed earlier, in suprarenal gland, what is present? In suprarenal gland, what is present? Outer cortex, inner medulla. Cortex is larger, medulla is smaller. We have discussed. So in the similar way, in kidney also, once you open the kidney, okay, by taking a coronal section, what you can see? Two things you can see. First, the outer part is the reddish-brown cortex, and inner part is the pale medulla. Okay, that you can see. Now, this is the. I hope you are familiar with this diagram. I know you are familiar with this diagram. You have seen this diagram somewhere, right? So this is nothing but the coronal section of the kidney. Okay, now in this particular section, what you can see? So you can see the cortex, outer part is the cortex, which is granular in appearance, and you can see the medulla, which is striated or striations are present in the medulla. Okay. Now this particular part is the capsule. Okay, this particular part is the capsule. Below the capsule, this outer part is made up of renal cortex. This outer part is made up of renal cortex. Okay. Now what is there in the cortex, that we will discuss. I am coming into that. Okay, once you open, what you can see, two things. First, the outer part is the cortex, inner part is the medulla. What is the appearance of the cortex? As I said, it is granular in nature. Appearance of the cortex will be granular. And what will be the appearance of the medulla? Striated. Striations will be there. So as you can see, this is the cortex and this is the medulla. Okay, this is the medulla. Okay. So these are the two things which you can see. Now you have to know what is exactly present in the medulla and what is exactly present in the cortex. This you have to know, because after that only we can go further into the microscopic structure. Okay. Now if you see the medulla, it is made up of what? It is made up of 8 to 10 medullary pyramids. Now what are those medullary pyramids? These are triangular areas of the renal tissue. Okay, which are striated, pale and conical structure. So if you concentrate on the medullary area, what you can see? First you can see 8 to 10 pyramids. Now these particular pyramids are conical in shape. That means they will be having one apex and they will be having one base. Okay, and if you see that particular pyramid, they are striated. Okay, now in case of pyramid, the base is directed towards the cortex. Okay, so the base will be directed towards the cortex and the apex or the papilla directed towards the renal pelvis. Okay, and fits into the minor calyx. Next, if you see the papilla in the apical portion of each pyramid, it projects into the minor calyx. Okay, so the pyramid, the apical part will project into the minor calyx. Okay. Now the striations which we see, just now I have mentioned that the pyramid which are there, in that you can see the striations. The striations which we see, that is mainly due to the U-shaped loop of Henle, collecting tubules, duct of Bellini, arterioles recti and vena recti. Okay, these are the structures which are passing through the renal pyramid. That's why the medulla, it appears striated in nature. Okay. Next, another important area is there, that is called area cribrosa. Now what is this area cribrosa? Now if you see the renal papilla, where the papillary ducts, okay, mainly the collecting ducts of Bellini, they open into the minor calyx. Okay, so numerous ducts are going and they are opening into the minor calyx. Okay, so the apex which is present, in that the opening is present, that is called papilla. Through that papilla, the ducts are going and opening into the minor calyx. Okay, so this particular structure is called as area cribrosa, and that allows the urine to drain into the renal pelvis. Okay, now let's go back to the diagram. So I was talking about this particular pyramid. So this is one renal pyramid, this is one renal pyramid, this is one renal pyramid, this is one renal pyramid. Okay, so if you see the diagram properly, these renal pyramids are conical shaped. Okay, so they are having apex. So this is the apex, this is the apex, and this is the base. Now if you see the apex, it is projecting into the minor calyx. Okay, and at the apex, the opening is there, that is called papilla, and through that papilla, the ducts, different ducts are draining into the minor calyx. This is how after filtration the urine is flowing, because from minor calyx it will go to the major calyx, the urine which is formed. From minor calyx, then major calyx, then renal pelvis, and from renal pelvis it will come into the ureter. Okay, this is the direction of the flow of the urine. That also you have to understand in the microscopic structure of kidney. Okay. Okay, next is the cortex. So renal cortex, what you can see, the outer part is made up of renal cortex. Now in renal cortex, mainly we are getting the renal columns and cortical arches. Okay, so what are those renal column? So just now I have shown you the pyramids. Okay, suppose one pyramid, another pyramid. In between the two pyramid, the area which is present, that is made up of cortex. Okay, and that is called as renal column. Okay, next, what are those cortical arches? So the tissue lying between the bases of the pyramid and the surface of the kidney, these are called as cortical arches. So the base of the pyramid, okay, above that we are having the area of the cortex, okay, that particular part is forming the cortical arches, okay. Now in cortical arches also the striations are present, and those striations are called as medullary rays, okay. In cortical arches also striations are present, and these are called as medullary rays, okay. Now what are those medullary rays? Now these are the conical mass where the apex is directed towards the surface of the kidney and base is continuous with the striations of the pyramid, okay. So from there the striations are continued and then it is reaching the pyramid, okay. Now if you see each medullary ray, it is made up of collecting tubules and duct of Bellini, and one medullary ray, and on either side we are getting the interlobular blood vessels, okay, that also reach the medullary ray. This is how the orientation of the cortex is there in case of kidney. Next, the tissue which is lying between the adjacent pyramid, that is called renal column, just now I mentioned, okay. Next, the cortical labyrinths, these are the region of the cortex between adjacent medullary ray. So one medullary ray, another medullary ray, in between that the region which are present, these are called as cortical labyrinth, okay. Next, the area of the cortical arch, okay, the area of the cortical arch on each side bounded by interlobar blood vessel, and in the central axis one medullary ray is present, that is called as the lobule of the kidney, okay. Now the area of the cortical arch bounded each side, okay. So cortical arch, on either side of the cortical arch what we are getting, interlobular blood vessel, and on the center we are getting single medullary ray. This particular structure is called as lobule of the kidney. Now here you can see, so this particular part is the cortex, this is the medulla, okay. Now in medulla I have mentioned that striations are present, okay. So here you can see, because of these structures the striations are present. Next, here you can see the medullary rays. So these are the medullary rays. Now if you take one medullary ray, okay, suppose you are taking this medullary ray, on either side we are getting interlobular blood vessel, okay. Now this particular structure will form the lobule of the kidney, okay. So first there will be cortical arch, okay. First there will be cortical arch, on each side we are getting the interlobular blood vessel, and at the center we are getting one medullary ray. That will be forming the lobule of the kidney, okay. Next, so that was about the coronal section of the kidney. In coronal section of the kidney we have seen cortex and medulla, what is present in the cortex, what is present in the medulla, that we have seen. Now we have to go inside the cortex, we have to go inside the medulla to see what exactly are present, okay. So for that we have to discuss the microscopic structure. Now inside the kidney, I don't know whether you have heard or not, approximately 1 to 4 million closely packed tortuous uriniferous tubules are present. Anyone, have you heard about the uriniferous tubule? Yes, I hope you have heard about uriniferous tubule, okay. So inside each kidney approximately 1 to 4 million, okay, closely packed tortuous uriniferous tubules are present. Now what are those uriniferous tubules? Okay, now this particular uriniferous tubule is made up of two parts, okay. What are they? First is the nephron, that is the secretory part, and the collecting part is also there, okay. So nephron is present and the collecting part is there. Of course the development is different, okay. So the collecting part and the secretory part, the development is not same, it is different. Now if you see the urinary tubules, they are held together by some amount of connective tissue, and in that connective tissue blood vessels, lymphatics, all those things are present, okay. So that particular connective tissue along with blood vessels, lymphatics, they are acting as stroma of the gland, okay. So now we'll go to the nephron, okay. I know already we have studied about the nephron earlier, okay. So today we will be discussing in detail about the nephron. Okay, so nephron is a structural and functional unit of kidney. Each kidney is approximately having 2 millions of nephrons. Now each nephron is made up of renal corpuscle and long renal tubule. So again in nephron also two parts are there. Okay, nephron is a structural functional unit. Approximately 2 million nephrons are there. Next, if you see the nephron, it is made up of two parts. What are they? Renal corpuscle and long renal tubule. Okay, so this renal corpuscle is also called as Malpighian bodies. What is the function of this renal corpuscle? Filtration. Okay, and then we are having the renal tubule, which is very long. And what is the function of the renal tubule? Selective reabsorption. Okay, so in nephron, how many parts are there? Two parts. What are they? Renal corpuscle and renal tubule, long renal tubule. Renal corpuscle का function क्या है? What is the function of renal corpuscle? Filtration. Okay, the blood which will come, it will filter it. And what about the long renal tubule? Selective reabsorption. Okay, have you studied renal physiology? Not yet. Okay, so in renal physiology, all those things, the functional aspect will be discussed. Now, if you see the renal tubule, it is made up of what? Have you heard about PCT, DCT, loop of Henle? Yes, I know you have studied all those things. Okay, so if you see the renal tubule, it is made up of what? As you can see, renal tubule is made up of PCT, loop of Henle and DCT. These are the structures which are forming the renal tubule. Okay, now the length of the nephron is approximately 50 to 55 millimeter. Now let me show you one diagram. Okay, I hope you have seen this diagram earlier. So this is the diagram showing the structure of, this is the diagram showing structure of what? Nephron. What you can see in this diagram? First you can see the renal corpuscle. See, this particular part is forming the renal corpuscle. And what about this part? This is forming the renal tubule, long renal tubule. ठीक है, नेफ्रॉन का दो पार्ट है, as I have mentioned. First is the renal corpuscle and long renal tubule. Okay, so this is the renal corpuscle. Okay, and this is the long renal tubule. In renal tubule, what is present? First is the PCT, proximal convoluted tubule. Okay, so this is the PCT, proximal convoluted tubule. Then what is this? This is the loop of Henle. And after loop of Henle, what is this? This is the DCT. Okay, so first what we have to study? We have to study about the renal corpuscle. What is the structure of renal corpuscle? Okay, now here you can see the structure of renal corpuscle also, where we have to know about the glomerulus, which are nothing but the tuft of capillaries. Okay, and the woman's capsule or the glomerular capsule. Okay, can you see the, what is this? And what about this capsule? This is the woman's capsule and this is the glomerulus. Now, can anyone tell me a peculiarity in this particular glomerulus? And glomerulus, we know that it is made up of tuft of capillaries, rounded capillaries. Okay, now what is the peculiarity of this particular capillaries? What is the peculiarity of this particular capillaries? Yes. See, you know that whenever a capillary bed is formed, first of all, the blood will come through arterial and capillary bed, and then blood will go through venous end. But here, we are having afferent arteriole and efferent arteriole, no venule. Okay, so the capillary bed which is formed, and the blood, when it is coming out, okay, going in through afferent arteriole, and from the capillary, when it is going out through efferent arteriole, not venule. Okay, so that is a unique property in case of the glomerular capillaries of kidney. Okay. Next, here we have to use one concept, and that concept already you have studied, and that concept is about the peritoneum. Just imagine, we have to use the concept of peritoneum which you have studied earlier. Okay, that also inside the woman's capsule. Okay, now do you remember the peritoneal folds which you have studied? After that, you have studied peritoneal cavity is there. Any organ which is invaginated into the peritoneal cavity, uska kya hota hai? It is having two layers. The inner layer is the visceral layer, outer layer is the parietal layer. In between two layers, what is present? Peritoneal cavity is there. Same concept will be followed over here. Okay, but names will be different, but the same concept will be followed over here. Okay. Now, based on the renal corpuscles, nephrons are classified into two types. What are they? These are the cortical or subcapsular nephron and juxtamedullary nephron. Anyone, have you heard about juxtamedullary nephron? Yes. Okay. So two different types of nephrons are there. Okay. First is the cortical nephron. Majority of them are the cortical nephron, or also called the subcapsular nephron. Okay. So in this particular case, the renal corpuscles are lying in the outer cortex. Okay. And if you see the loop of Henle, they have short loop of Henle that extend only into the outer part of the medulla. Next, in case of juxtamedullary nephron, their corpuscle lie at the junction of the cortex and the medulla. Okay, so the placement is different, and obviously function will also be different. Okay, now if you see the juxtamedullary nephron, they help in maintaining the concentration of the urine. Okay, so mainly the concentration of the urine is maintained by the juxtamedullary nephron. Okay, so in kidney, in case of nephron, I have mentioned that two parts are there. Okay, renal corpuscle and long renal tubule. Okay, so first we'll discuss about the renal corpuscle. Okay, so what is present in the renal corpuscle? Renal corpuscle is also called as Malpighian body. Okay, so here you will be getting a round tuft of blood capillaries that is called as glomerulus, and the cup-like double-layered covering glomerulus called as glomerulus capsule or Bowman's capsule. Okay, so two components are there. Tuft of capillaries are there, which is called as glomerulus. Rounded tuft of capillaries, that is called as glomerulus. Okay, and that is covered by a double-layer cup-shaped structure that is called as Bowman's capsule. Okay. Now, the glomerular capsule represent the cup-shaped blind beginning of the renal tubule. So the Bowman's capsule which is there, okay, from there only the renal tubule starts. That means the PCT will start from this particular blind beginning, okay, which is cup-shaped. Next, between the two layers of the capsule, there is a space, potential space, that is called the urinary space. Okay, now what is this two shaped, so as two layers? So as I said, the tuft of capillaries which are there. Now this particular tuft of capillaries, they are present inside the cup-shaped capsule. So this cup-shaped capsule which is present, okay, actually tuft of capillaries are invaginating. So though it is invaginating, that's why two layers are present. The innermost layer which is attached to the, or which is very near to the capillaries, that is the visceral layer, and another layer is there, that is the parietal layer. Okay. Now if you see the diagram, I hope you have seen this kind of diagram. Now in this diagram you can see what is this. These are the tuft of capillaries. Okay. Now here you can see the blood is coming through afferent arteriole and leaving through efferent arteriole. Venule are absent over here. Okay. Now this particular tuft of capillaries, you can see this is invaginating inside a cup-shaped capsule that is called as Bowman's capsule. Now once it is invaginating, just apply the concept of peritoneum which you have studied. Okay, so you will be getting two layers. Can you see the two layers? See, this is one layer, this is the visceral layer, and this is another layer, this is the parietal layer. In between these two layers, the space which is present, this is called as the urinary space. Okay, that is called the urinary space. Next, this particular Bowman's capsule which is there, okay, now it is in this particular part, it is beginning as the long renal tubule. Means it starts, or you can say it gives the origin of the long renal tubule. Okay, from here you can see the PCT, proximal convoluted tubule, starts. Okay, now two layers are there, as you can see. So the outer layer is mainly lined by mesangial cell. Here it is called as mesangium. Okay, and the inner layer which is present, it is lined by a special type of cell, and these are called as podocytes. These are called as podocytes. Why they are called as podocytes? Because they are having foot-like processes. Okay, and these are called as pedicles. That's why these cells are called as podocytes, and they are very important. Okay, the filtration which is taking place from the blood okay, this particular podocytes are playing an active role in the filtration. Okay, so in case of glomerulus, is a lobulated tuft of anastomotic fenestrated capillaries, okay, where afferent and efferent arterioles are there, which will project into the Bowman's capsule. Okay. Next, blood enters through tuft of afferent arteriole and leaves through the efferent arteriole. Here, the venula absent. Okay. Next, now the afferent and efferent glomerular arterioles, okay, they are forming a particular part, and that is called as the vascular pole of the renal corpuscle. Okay, so the tuft of capillaries which are present, they are forming the vascular pole. Now each lobule consists of capillary loop, and that is suspended in the capsular space by a mesentery from the visceral layer. So see, here also mesentery is present. I hope you remember the folds of intestine. Do you remember the folds of intestine, small intestine? They are supported by what? Peritoneal fold, that is called as mesentery. So here also, just now I have shown you tuft of capillaries are there. Those capillaries also need a support, and that support is provided in the form of mesentery, and that is called as mesengium. It is called as mesengium. Okay. So here you can see the mesentery contain mesengium, that is made up of mesangial cells surrounded by mesangial matrix, which is supporting the tuft of capillaries. Okay. Now this mesangial cells very important, okay, because they contain filaments similar to myosin, and they bear angiotensin II receptors. Okay. Now this mesangial cell play a very important role in controlling the blood flow through the glomerulus. Okay, so those mesangial cells which are supporting the tuft of capillaries, they control the blood which is flowing through the capillary bed. Okay, now mesangial cells also have important function in phagocytosis, support the glomerulus, and synthesis extracellular matrix. So these are some functions of the mesangium which is present in that particular area. Okay, and this particular mesangium becomes very prominent in glomerulonephritis. Okay, so here you can see the diagram. This is a beautiful 3D diagram, okay, of a renal corpuscle, where can you see this is the afferent arteriole. So see, blood is entering like this. Now here the capillary bed is present, and this is the efferent arteriole through which the blood is coming out. Okay, next I am talking about this particular area. Okay, so see this particular area where the mesangium is present, or you can say it is similar to the mesentery. So this tuft of capillaries which are present, this is supported by this glomerular mesangium. Okay, it is supporting, it is producing extracellular matrix, it is regulating the flow of blood through this particular capillaries. Okay, okay. Next is the glomerular capsule or the Bowman's capsule. So already I have shown you the capsule or glomerular capsule. Okay, it is dilated blind end of the renal tubule which is invaginated by glomerular plexus. Now if you see the capsule, double layered cup consists of outer parietal layer, you can see outer parietal layer and inner visceral layer. Okay, and two layers of which are separated by a potential space that is called as urinary space. Just imagine the peritoneal cavity, okay, and here it is called as urinary space between the two layers. Now if you see the parietal layer, it is lined by flattened epithelium resting on the basement membrane, and the visceral layer is lined by discontinuous polyhedral cell and called as podocytes. Very important. Okay, so that was absent in case of peritoneum. Okay. Here two layers are present and a potential space is there which is called as urinary space. Outer layer is lined by endothelium. Okay, and if you see the inner layer, a special type of polyhedral cells are there, and these are called as podocytes. Okay. Now the urinary space become continuous of the renal tubule at the urinary pole. Okay, so the part from which the PCT start, that particular part is called as urinary pole. Okay, now already I have mentioned why these are called as podocytes, because they possesses foot-like processes. Okay, because of the presence of those foot-like processes, they are called as podocytes. Now if you see the podocytes, first of all they are having numerous tentacle-like major primary processes parallel to the basement membrane. Now each major or primary process again having numerous secondary processes, these are called as pedicles. Okay, so two things are there, major or primary processes are there. From that major primary process again numerous small pedicles are there. Okay, now those cell body of the podocytes will come in contact with the basal lamina through these pedicles. Okay, now the pedicles of the adjacent podocyte will interdigitate to form the filtration slit. Okay, so those pedicles will interdigitate with each other to form the filtration slit. Okay, and this particular slit is covered by slit diaphragm through which the filtration is taking place. Okay, let me show you the diagram. Yeah, I hope you can see the diagram. So this is how the podocyte look like. Okay, this is how the podocyte look like. So you can see this is the cell body of the podocyte. After that you can see these are the primary processes. Okay. Now from the primary processes you can see these are the secondary processes, or these are also called as pedicles. Okay. Now this pedicle, what it will do, it will go and attach to the basal lamina. So you can see this is the basal lamina. Okay. And what is this? These are the endothelial cells of the capillaries, the tuft of capillaries. Okay. Now this particular pedicles, okay, they will interdigitate with each other to form the slit, filtration slit, and over the filtration slit a slit diaphragm is there. Through that slit diaphragm only the filtrations are taking place. Okay, that's why podocytes plays a very important role in the filtration. So whatever blood is flowing through this particular blood vessel, okay, the filtration will be taking place by this podocytes only. Okay. Next, there are some areas of the capillaries which are not covered by podocytes, where the podocytes are absent. In that case, mesangial cells are there. Okay, and from that particular part, filtrations are not taking place. Okay, जहाँ पे podocyte है, from that particular part only the filtration will take place. Next, so just now I have mentioned about the basement membrane. Now what exactly is that basement membrane? This basement membrane is called as glomerular basement membrane. Okay. Now it is a very thick basement membrane. If you see the thickness, it is approximately 300 nanometer. Okay. Now this particular basement membrane act as a filtration. Now glomerular basement membrane, as I said, it is a very thick basement membrane, approximately 300 nanometer thickness. Okay, now that particular glomerular basement membrane is very important because it acts as a filtration barrier that will separate the urinary space from the blood capillary. So just now I have mentioned that a potential space is that is called urinary space. Now that particular urinary space and the blood capillaries, they are separated from each other by this particular basement membrane. Okay, now what are the layers present in that basement membrane? 3 layers are present in that basement membrane. Okay, first of all, a electron dense layer that is called as lamina densa. Okay, and inner and outer layers, these are called as lamina rara interna and externa. Now if you see the lamina densa, what is present in that particular layer? The collagen fibers are present, that also type 4 collagen fibers, which act as a physical barrier. Next, electron lucent layers are there, in which glycosaminoglycan, heparan sulfate and present, which act as negative charge. Okay, so this particular glomerular basement membrane is a physical barrier and an electrical charge barrier for large molecules. Now why this particular basement membrane is present? Because see, if that particular basement membrane is not present, then when the filtration will take place, okay, through that important molecules or larger molecules can escape and then go into the urinary system, and then it will be thrown out of the body. Okay, now it is placed in such a way that, okay, it is a physical barrier, along with that it is also a electrical barrier. Okay, now this glomerular basement membrane, because of that physical and electrical property, it is acting as ultrafilter. Okay, now that will allow the filtration of water, simple solutes of blood plasma into the capillary space. Okay, so the filtration is taking place. Now filtration of what is taking place? That is allows the filtration of water and simple solutes of the blood plasma into the capillary space. Okay, now this filter consists of blood plasma without protein, lipid droplets and blood cells. Okay, that's why this glomerular basement membrane is very important, or else this particular molecules will also come out from the blood during the filtration. Now, if a molecule having size of more than 4 nanometer, it will unable to pass through this membrane. Okay, so you can see macromolecules greater than 4 nanometer cannot pass through the filtration slit. That is also very important. Okay, now this glomerular basement membrane and podocyte is not present all around. So in that particular part where these podocytes are absent, in that we are getting mesengium. Okay, now this glomerular filtration barrier, as I said, separates the blood capillaries from the urinary space. Now here is the diagram. Okay, in this diagram you can see the glomerular basement membrane. So this is the glomerular basement membrane. Okay, so this is the blood capillary through which you can see the blood is flowing. Okay, now what are this? These are the podocytes which are present in the inner layer or the visceral layer of mesengium. Okay, now I hope you can see the processes of the podocytes. Okay, so from there only the filtration will take place. Okay, and here you can see the glomerular basement membrane. Okay, now this glomerular basement membrane is important, as I said, because of the 3 layers which act as a physical barrier, and electron barrier is also there, that means charge barrier. Okay, so what it will do, it will not allow the important molecules to escape the filtration slit. Okay, that means any molecule having size more than 4 nanometer will unable to pass through this slit. Okay, next I have mentioned all around the capillary you cannot see the podocytes. So where the podocytes are absent, you can see the mesangial cells are present. Okay, so from this part the filtration will not take place. So that is all about the renal corpuscle. Next, after the renal corpuscle, what is this? That is the renal tubule. Okay, now in renal tubule, what are the parts present? First is PCT. Okay, then loop of Henle and DCT. Okay, now there is a chart. Okay, you can see this is a chart showing the difference between the PCT and DCT. Okay, now this chart is mainly based on the histological appearance. When you will see the slide under microscope, especially when you will be concentrating, at that time there will be so many cut section. At that time you have to identify which is PCT, which is DCT. In the diagram it is very easy to identify, but under microscope how will identify? For that there are some important features which you should know. Okay, so in case of PCT the length is longer, in case of DCT it is very short, only 1/3. Okay, next if you see the diameter, it is wide, it is narrow. Okay, so this is also important identifying feature which will be helping you to identify the PCT and DCT under microscope. Okay, next you can see the epithelium, it is simple cuboidal to low columnar. Here only you are getting the simple cuboidal epithelium. Okay, next when we are using H&E staining, at that time this DCT is dark pink stain than DCT. That means this particular PCT will stain more darker as compared to the DCT. DCT will be stained lighter. Okay, so PCT is dark stain, DCT is light stain. This is also important feature to identify between the PCT and DCT under microscope. Okay. Next, if you see the nuclei in cross section of the tubule, it is fewer, more equally spaced. Next, if you see the lumen of the PCT and DCT, here it is star shaped irregular. In case of DCT, it is circular, oval or regular. Okay. Now the cells which are present, okay, the cells which are present inside the PCT and DCT, a difference is there. What is the difference? Here the brush border is present, here brush border is absent. Now, what is the meaning of that brush border? Microvilli. Okay, already I have mentioned several types. 2 types are there, brush border and striated border. Here we are having brush border. So that brush border is present over here, it is absent in case of DCT. Okay, these are some important points which you should know in order to identify the difference between PCT and DCT under microscope. Okay, so one is darkly stained PCT, DCT is lightly stained, PCT is wider, DCT is the lumen is narrower. Okay, here we are getting the simple cuboidal to low columnar, here only we are getting the simple cuboidal epithelium. Okay, so these are some important points. Next, can anyone tell me what are the functions of PCT and DCT? Yes, selective reabsorption. Very good. Specifically, can you mention? See, in case of DCT, mainly the sodium ions, then bicarbonate ions, all these are absorbed. Okay, after that, so that the urine becomes acidic in nature. And what about the PCT? Electrolyte absorption, amino acid absorption. Okay, those absorption are taking place, which is making the urine concentrate. Okay, so this particular PCT is helping a lot to make the urine concentrate. Okay, approximately 80% of the reabsorption is taking place in the PCT. And if you see the PCT, it is coiled shape. It's a straight nahi rehta hai. It is in the form of coil. Okay, so that's why the length is more time is there, so more absorptions are taking place from PCT. Okay, so PCT and DCT is done. What is the next? Next is the loop of Henle. Now, in case of loop of Henle, please note that the loop of Henle which is present, it is present along with the cortical nephron. It is also present along with the juxtamedullary nephron. Okay, so both aspects you have to study in the loop of Henle. Okay, so that's why I have seen students used to get confused, but both the nephrons are having this loop of Henle. Now, loop of Henle already you know, it is U-shaped, having a descending limb, then the loop, and then the ascending limb, which is thick. So descending limb generally it is thin, ascending limb it is thick. Okay, and in between we are getting the loop, or also called as hairpin band. Now the descending limb, if you see, it enters the renal pyramid as a continuation of proximal tubule. Okay, so this particular descending limb will enter the pyramid, renal pyramid. Now the loop of, in case of juxtamedullary nephron, reaches the apical zone. Okay, where in case of cortical nephron reaches the basal zone of the pyramid. That is the difference between the cortical nephron and juxtamedullary nephron. Now the descending limb of the loop constitute the thin segment where the diameter is only 15 micrometer. Okay, and in case of thin segment it is lined by simple squamous epithelium. Next, the ascending limb, okay, is making the thick segment where the diameter is 30 micrometer, and here you can see it is lined by cuboidal epithelium. Okay, so the thin segment is lined by which epithelium? Squamous epithelium, and thick segment diameter is double and it is lined by cuboidal epithelium. Okay, next the loops are also surrounded by arteriole rectae, vena rectae. Okay, and these are called as, this is also called as ansa nephroni. Okay, next, what is the function of the loop of Henle? 5% of the fluid absorbed. Approximately 5% of the absorption is done by loop of Henle. Okay, now ascending limb will pump out the chloride and sodium ion passively into the tissue space. After that, what will happen? Descending limb diffuses water and some chloride and sodium enters the loop. Okay, like this, what it will do? It will maintain the graded osmolarity in interstitial fluid of the pyramid. Okay, anyone, have you heard about the counter current mechanism of the kidney? Okay, so that counter current mechanism is mainly maintained by the loop of Henle. So about the counter current mechanism you will study in detail in physiology. Now here you can see this is a beautiful diagram where entire structure of nephron can be seen. Okay, now here you can see the renal corpuscle. Okay, these are the tuft of capillaries, these are the, you can see, afferent, and this is the efferent part. Then what is this? This is the Bowman's capsule. So in Bowman's capsule you can see two different types of cells are there in two different layers. After that, from here you can see the renal tubule starts. Okay, so this is the PCT. See, this is the PCT. Okay, then this is the loop of Henle. Now this loop of Henle you can see mainly it is present. See, two different types of nephrons can be seen over here. Okay, this is the cortical one and this is the juxtamedullary one. Okay, so the loop of Henle is related to both, that you have to understand. Okay, as you can see, it is related to both. Okay, next here you can see this is the which limb? Descending limb. This is the loop and this is the ascending limb. So ascending limb is thicker, descending one is thin, and then you are having the loop. Now this particular part Okay, this particular loop of Henle, as you can see, it is forming the counter current osmotic, or you can say counter current mechanism of the kidney, okay, which helps in reabsorption. Okay, then this is the ascending part, and then this is the DCT. So DCT is very short as compared to the PCT, as you can see in the diagram. Okay, next is the collecting tubule. Okay, so the collecting tubules, if you see the diameter 40 to 50 micrometer in diameter, they begin in the cortex as a continuation of DCT. Okay, so the collecting tubule, how it is formed in the cortex, that is nothing but the continuation of the DCT. Okay, proceeded to the medullary ray. Now in medullary ray, the collecting tubules, okay, multiple collecting tubules will join to form the collecting ducts. Now these collecting ducts run towards the apex of the pyramid. Okay, they will run towards the apex of the pyramid. So the pyramid which is present in the medulla of the kidney. Okay, now they will join to form the papillary duct of Bellini. Okay, now this duct of Bellini opens into the free surface of the papilla. So papilla is present at the apex of the pyramid, which I have mentioned in the beginning. Okay. Now both the collecting tubule and the duct of Bellini, they are lined by simple cuboidal epithelium. Okay, so from duct of Bellini, which is there, okay, where it will open into the papilla. Now where the papilla is present, at the apex of the pyramid. Okay, from there it will go into the minor calyx. Okay, so cells of the collecting duct do not have microvilli. Okay, and the duct of the Bellini are surrounded by hypertonic tissue fluid, which is present in the renal pyramid. Now this collecting tubules, they convey and modify the ultrafiltrate from the nephron to the minor calyces. Okay, from minor calyx, major calyx, from major calyx the ultrafiltrate will go to the renal pelvis. From renal pelvis it will go to the ureter. Okay, this is the direction of the flow of the urine. Now this is the diagram. Okay, so I have mentioned the various part of the renal tubule, right? Now if you see the lining of the renal tubule, okay, multiple different types of cells are there. In each and every part we are not getting the same lining epithelium, it is different. Okay, so here you can see first, what is this? This is the PCT. So you can see the cells, simple cuboidal epithelium, and here you can see brush border are present, that is the microvilli. Okay. Next, this is the straight tubule. Here also you can see simple cuboidal epithelium with brush border. Next, this is the thin segment of the loop of Henle. In case of thin segment, as I mentioned, what is the lining epithelium? Simple squamous. Okay, so as you can see, this is the simple squamous. Next, simple cuboidal epithelium in case of thick ascending limb, where brush border are absent. Here also you can see simple cuboidal epithelium in DCT, brush border is absent. Okay. Next, simple cuboidal, especially in case of collecting tubules, duct of Bellini, all are lined by simple cuboidal epithelium. Okay. So this particular diagram will help you to understand the lining epithelium of various ducts which are present in the renal tubule. Next, this is a comparison chart where you can see same the lining epithelium which are present at various segments, the length along with the diameter. Okay, so first you can see the woman's capsule, okay, where flattened epithelium are present, and along with that another special type of cells are also present, those are called as podocytes. Next you can see the PCT, thick descending segment, you can see thin descending segment, hairpin bend, then thick ascending segment, DCT and collecting duct, and then you can see the lining epithelium. Okay, just now I have showed you in the diagram. Then the length of each and every part, and then the diameter of each and every part. Okay, length are given in millimeter, diameter are given in micrometer. Okay, next another important part which is related, that is the juxtaglomerular apparatus. I hope you have heard. Now don't compare or don't mix it with juxtamedullary nephron. So juxtamedullary nephron and juxtaglomerular apparatus, both are totally different. It is not same. Okay, I have seen students used to do the mistake. Okay, both are totally different. Now if you see the juxtaglomerular apparatus, mainly three components are there: macula densa, juxtaglomerular cells and Lecis cells. Okay, Now this particular apparatus, first of all, the juxtaglomerular cells are there. Now what are those juxtaglomerular cells? Now these are the modified smooth muscle cells which are located in the tunica media of the afferent arterioles. Okay, so if arterioles are there, tunica media will be there. Tunica media में क्या रहता है? Smooth muscle cells are there. So it is the modification of the smooth muscle cells of the tunica media of the afferents. Okay, now they are situated very close to the medulla. Now what is the function of this juxtaglomerular cell? They will secrete renin. Okay, and this act as a baroreceptor, reacting when there will be fall in the blood pressure, and then they will release the renin. Okay, now secretion of the renin is also stimulated by low sodium level in the blood and by the sympathetic stimulation. So that is the function of juxtaglomerular cells. Okay, now here you can see the juxtaglomerular apparatus. Okay, so here you can see juxtaglomerular cells are present, and you can see the cells of the macula densa. Now macula densa, these are the modified cells of the DCT near the afferent arteriole. Okay, where in close contact with the afferent arterioles, where the basement membrane of the tubule disappeared. Okay, the lining epithelium first of all densely packed together and are columnar rather than the cuboidal. Okay, now these are called macula densa cell. Now what are the functions of this macula densa cell? They will lie very close to the juxtaglomerular cells. Okay, now this particular macula densa cell, they will monitor the ionic concentration in the blood. Okay, ionic concentration of the fluid passing across the tubule. Okay, so after filtration, the ionic concentration which is present in the tubule, that is monitored by this macula densa. Okay, and if required, they will make the juxtaglomerular cell to secrete renin. That is the main function of the macula densa cells. Okay, lastly, the mesangial cell are also called as vesical cell. Okay, these have lace-like structure. Okay, so and they are having angiotensin II receptor. Okay, and they probably regulate the glomerular filtration rate. Now exact function about the mesangial cell or the lacial cells are till unknown. Okay, so that is all about the juxtaglomerular apparatus. Next, I have mentioned about the two types of nephrons, that is the cortical nephron and juxtamedullary nephron. Okay, so here you can see two different types of nephrons. Okay, cortical nephron and juxtamedullary nephron. So I will request everyone to go through it. Okay, so that you can understand the differences between the these two nephrons. First of all, where is the location? Okay, what is the important function? What is the rate of filtration? Okay, that is mentioned over here. Okay, lastly, let me show you the microscopic diagram. So this is the schematic diagram which you should draw. Now in this schematic diagram you can see the renal capsule. Okay, this is the cortex. In the cortex mainly you can see the glomerulus. See, these are the glomerulus. The tuft of capillaries are also there. Okay, and then when you will see the medulla, in the medulla you can see all the ducts are present. Okay, so this particular part specially we will be discussing in histo class. Okay, now I have explained you about the microscopic structure. So the microscopic diagram, where what you will see in case of cortex, what you will see in case of medulla, that will be discussed in the histo class. Okay, okay, so that's all everyone. Okay. Yeah, these two things, this will be discussing in the histo class along with the slides. Thank you everyone.